Well, the last topic is color vision, and there's a few main points that I want to cover that I think students have a hard time really grasping, and I want to make sure that we're teaching correct principles. And we'll start with a question about which depiction of vision is correct. Now, in the top view, as this woman is viewing this green square, okay, there are arrows from the square to the eye demonstrating, the transmission of some type of energy or something from the square to her. The second depiction is that from her eyes something is being emitted, or there's some type of energy that is allowing her to view or see the green square. And so do you believe that this is the correct view of how we see, that this is the correct view, or is it some of both? Okay, so just think about that for a minute. And I'm not going to have you publicly answer this one. I mean, you're welcome to talk about it with your neighbor and see what they think. Okay. The correct answer is A. Correct answer is A. Okay. In terms of vision, we're not doing anything. Okay. I mean, yeah, we open our eyes, but nothing is coming out of our eyes. Okay. It's all coming in, and it's simply electromagnetic energy. Now, in a study by Weiner in like 2000 or so, they published about half of American adults believe that at least some element of this is occurring, that somehow we're actively seeing something that comes out of our eyes, or at least the way they phrase the question. So first clarification is, nothing's coming out. It's whatever comes in, and those are of course light rays, type of electromagnetic energy. In terms of how light and color work, first of all, let me just say that color does not exist in the world. Okay? Try and tell them that. Color only exists up here. Okay. In the world, okay, now there are some things that do exist in the world that are represented in our vision that hopefully we get an accurate representation of, that there actually is space in the real world. Okay? It is three-dimensional, okay? Even though what ends up on our retina is not. Okay? We have to recreate that. But color does not exist in the world. The only thing that exists are different wavelengths of electromagnetic energy. Okay? And a small sliver of those wavelengths can our eyes detect. Okay? Are we sensitive to? Now, in terms of why we see different colors, of course that happens all in our brain, but objects that we perceive to be different colors reflect, okay, unless they're a light source, okay, they are reflecting light into our eyes that varies in the spectrum of wavelengths that are being reflected. So a gray sheet of paper, and this is the visible spectrum of the electromagnetic spectrum, from about 400 to about 700 nanometers we perceive and can detect. A gray sheet of paper will reflect evenly all the different wavelengths. Okay? Similar to a white source of light, okay? All the wavelengths are present. They don't add together somehow. They all are independent, but they all come together, okay, in parallel. They all reach the eye. So it's gray because it equally activates all the different receptors that we have in our eye. A lemon appears to reflect some of the medium wavelengths of light that, in terms of color, if we only saw those wavelengths, we would perceive as green or yellow. A little bit of the longer wavelengths, kind of in the yellow region, and it also reflects a lot of the longer wavelengths, including wavelengths that we would perceive as red. Now, in the end, the color that we perceive a lemon to be is primarily yellow because it reflects a lot of this medium wavelengths of light. A tomato we perceive to be more red, though, because it reflects primarily only the very long wavelengths that we can perceive. It does reflect a little bit of what we might see as maybe orange. Okay? But for any object that we're perceiving the color, okay, it's reflecting a broad band, okay, a band of wavelengths of light. that will impinge upon just 3 different types of receptors in the eye, the cones. Okay? So where does color exist? Okay. Literally only in the eye or brain of the beholder. Okay. In fact, you've probably wondered, and you probably get asked this. Do I see the same color of blue that you see, when we look at the same object? You know the answer to that? Okay, so she says, well, my blue could be a little bit bluer, yours could be a little bit greener. And the only way that we would know that, actually, would be to do a color matching experiment. I give you a standard color, and then you mix red, green, and blue light in different intensities until you think it's the same as the standard. And we find that when humans do that, they have a wide agreement, although there are small differences, okay, between individuals. Okay? And certainly if you have some color vision deficiency, you're going to be way off on some colors. But it's technically not color blindness because they are not blind to colors. Okay? They can perceive colors. They just have difficulty discriminating certain wavelengths from each other because of the lack, typically, of a type of cone. So with color matching experiments, if I give you the blue and we ask people to adjust the intensity of a red, green, and blue light, we can match it. And this is where I think the myth starts to be perpetuated, is that suddenly, okay, because we can match any color with mixing just 3 colors of light, the myth begins that, no, we can only see 3 colors of light, okay, or only detect 3 colors of light. Is that true? No. We can detect a full range of colors, but we'll see just how that happens momentarily. Okay? So the first thing I want to say is that we don't detect just 3 colors of light. It is true that we can replicate any color that we're viewing by mixing only 3 colors of light, but that doesn't mean that we can only detect 3 colors of light, because that implies that we can only see a pure red, a pure blue, and a pure green, okay, which is not true. We can discriminate millions of colors, okay, until you get into the Home Depot paint store. I don't know, it looks the same to me. Okay, and I've got about 7 color names that I use in my repertoire. Red, orange. Okay, nothing fancy. Okay, which is why I have to ask my wife if that tie matches that shirt, right? Okay. All right, this shows the responsiveness of our 3 types of cones in the retina to different wavelengths of light that nicely have been colored in the background, right? So we can kind of see what we're talking about. Ignore this chart for the rods, okay, because they don't see color, right? They can detect light of medium wavelengths optimally, but they don't see color as we're going to talk about it. So just ignore that one. But otherwise, note that we have 3 different types of cones, each that respond maximally to a particular wavelength of light, but note that they respond to a broad spectrum of wavelengths. So we like to call these the blue cones. It's probably a bad name because they're not actually colored blue. Okay? If you look in the retina, they're not blue dots in there. Okay? But they do respond best to short wavelengths of light. So we call them the S cones, short wavelength cones. But note that not only do they respond to short wavelengths of light that we perceive as violet, okay, or indigo, or blue, okay, but they also respond some to those medium wavelength light. Okay? They respond to a broad range, but they respond best to these short wavelengths. There's another type of cone that responds best to the medium wavelengths of light. We call those the M cones. Okay? Probably shouldn't call them the green cones because it implies that they're green, but they're not. They respond best to the medium wavelengths of light. But note that the long wavelength cones, sometimes called the red cones, also overlap a lot with wavelengths that the medium wavelength cones respond to. But they respond better to either than any of them to those long wavelengths.
Are they known as L?
Yes, L. Very good. S, M, and L. Okay, I like them because it's easy. Okay.
And is this trichromatic?
Yes.
This is, right?
Yes.
So this is a much better explanation.
Okay. So back here, it was Young and Helmholtz, or at least Helmholtz. I don't know if Young did this part, but the color matching 100 years before we ever knew that there were 3 types of cones in the retina. Okay. He said, well, you know what? If we can match it with just 3, maybe our eyes only have 3 different types of receptors. Okay. And the relative activity of those 3 will determine the difference in what we perceive as color. So that's kind of, the color matching led to the theory, even though it wasn't anatomically shown for, I think, like another 100 years or so.
Wow, okay.
It's a long time. So it's pretty impressive reasoning, actually. Okay, any questions so far? Okay. So why are tomatoes red? Okay. Note that they reflect a lot of these longer wavelengths. But it's not any one pure wavelength. Only in the lab do you deliver pure wavelengths of light. Okay? I mean, like laser stuff, right? But in the real world, there's a broad range of wavelengths that are being reflected. But which of the 3 cones will be activated most strongly? The L cones, the long wavelength cones. It'll probably also activate the medium wavelength cones to some extent. And probably not much, if at all, any of the short wavelength. So now we have a lot of the L, some of the M, and none of the S. Okay? What's the only place on this chart, okay, that what color would we see? It would be something red, right? There's no other place on this chart where you can get high activity in the L cones, a little bit in the medium wavelength cones, and none in the short wavelength cones. So we're going to see red. Okay, that's why tomato is red, because it reflects longer wavelengths of light, which more strongly activate the long wavelength cones.
Is it really none of the S cones, or just very?
Probably hardly any. Okay. The other thing is that there's a lot more of the medium and long wavelength cones in the retina than there are of the short wavelength cones. Now, to help emphasize, this is one activity that you can do. Okay, I've tried it in other contexts. I haven't used it a lot, but I could divide the class up into 3 different types of cones. I could say, okay, you are the long wavelength cones, and whenever you see a stimulus that is reflecting longer wavelengths of light, I want you to indicate your responsiveness to that by raising your hand, super high if it's, wow, there's a lot of long wavelengths, or, there's maybe a little bit of long wavelengths or no wavelengths, okay? So you would be the long wavelength cones, you would be the medium wavelength cones, and you would be the short wavelength cones. So if I show you this square, raise your hand to indicate your responsiveness to the wavelengths of light that you think are in that. Okay, so if there's a lot of long, you should be way up there. If you think there's a lot of medium, your hand should be way up there and keep them up. And if there's any short wavelength cone in this middle square, then raise your hand. So everybody do that right now. Okay. All right. So I'm seeing a lot of long and medium, but no short. Okay. And without even looking at the screen, I'm going to guess that you're looking at yellow. Okay. All right. Let's try another one. Okay. And how about this one? Ooh, there's an even responsiveness, but not particularly strong to any of them. It's probably gray. Okay. I can do this without looking. Okay. And what's this one? I'm guessing that it's a blue color, right? So I'm the brain having to determine, what color am I going to create based on the relative activity of the 3 different types of cones. Okay? And that's all it is. So true or false? The trichromatic theory states that our eyes are sensitive to only 3 colors of light. Absolutely false. Okay? Absolutely false. Okay. Another thing gets really confusing is additive and subtractive color mixing. Okay? It's just like positive and negative reinforcement, right? I mean, it's like, additive and subtractive. Okay. Subtractive color mixing is probably best illustrated by using colored cellophane over a pure light source. okay? Because as you add each layer of cellophane, okay, whatever color it is, it subtracts out the other colors. Okay, it only lets yellow pass, but not the short wavelengths or the really long wavelengths. Okay, so that's probably a great example, the best for subtractive color mixing. But it's also done with paint. Oh, first of all, why when you talk about the trichromatic theory color vision, you say so. You can replicate any color by mixing red, blue, and green light. But somebody inevitably is going to say, but wait a minute, those aren't the primary colors, right? Well, how come? Because we're talking about mixing light, which is additive color mixing, versus mixing pigments or paint, which is subtractive color mixing. Okay? So in a printer's shop, what colors do they use to print? Actually, it's a little more complicated. They actually use a cyan, magenta, yellow, and black, right? It gets a little complicated. It's like, it's even more complicated. But red, yellow, and blue are the primary colors for mixing paint, basically. And I remember as a kid learning that and thinking, oh, I'm going to create a new color of paint, right? So it was like finger painting age, right? And so I started mixing all these different colors of paint together, thinking I'd get some new color, and what I end up with. Yeah, brownish black, just, ooh, that's ugly, right? That didn't get any prettier by adding more colors to it. So, because I didn't understand subtractive color mixing. So, let's start with white light, which contains all the wavelengths of the visible spectrum. And, this is using filters, but I'll explain it with paint too in just a minute. If you have a yellow filter, it pretty much just allows those medium and long wavelengths to pass through. Okay? It blocks out the short wavelengths. A blue filter will allow the short and a little bit of the medium wavelengths to pass through, but it will block out those longer wavelengths. So when you combine these two filters together, the only rays that get through both of them are in the middle wavelengths, which we perceive as green. Okay? So this is subtractive color mixing because each stimulus subtracts out some of the wavelengths of light, and what you get at the end is what's common to all of them. So with paint, okay? Blue paint reflects a lot of the short wavelengths, but not much of a little bit of the medium, but not the long wavelengths. Okay? It absorbs those. Yellow paint absorbs the blue wavelengths, but it reflects the medium and some of the long. So the only wavelengths that get reflected from both are the ones in the middle, which we perceive as green. Okay? Because the blue is absorbing the yellow is absorbing the blue. The only thing that gets reflected from the mixture is green. Okay? But that's called subtractive color mixing because you're taking away wavelengths of light when you put things together.
And why, I always have trouble understanding this with color blindness. And why is green the most likely color that people who are colorblind are blind to?
Because the most frequent genetic problem with red-green color blindness is you are lacking either the medium wavelength cone or the long wavelength cone. So what happens is instead of having two slightly different cones to tell you the difference between red and green, you only have one cone. And on that cone's activity, there's actually 2 points that produce the same response. One of them is a red, and one of them is a green. So they confuse them. So what do they see? I don't know. I don't have that problem. We can look at pictures, okay, side by side that they can't discriminate the difference between them, but we say, whoa, that one looks so weird, and that one looks normal. Okay. In fact, you can submit images online, and it'll transform the colors in them to show you an image that a person with color vision deficiency would be unable to discriminate from the original. But the rest of us can say, whoa, that looks really different. So you can kind of get some glimpse into what their world is like. Okay.
My dad had red-green color blindness, and one of the things he described to me actually, he used to play the hunting video games, the really realistic ones with the deer in the forest, and he could pick out those deer so much better than me because the modeled greens of the trees weren't modeled to him. They were one like he explained it as they're one shade, and to me that brown sticks out because I don't see all the variation, I just see it as one. And so that helped me a little bit to understand it better, and I try to explain that to my students, and like, oh. And then of course there's always a couple kids when you put the Ishihara up and go, uh-oh, I don't see the number. I'm like, ooh, why don't you get those checked?
Yeah. Refer them to the right source.
Yeah,
Right?
exactly. Go.
Like, well, just could be a thing on the internet or my screen, or you could have a problem,
right. Just, just check it out.
So then additive color mixing is when you combine wavelengths of light. And here we're combining some short wavelength, the blue, the medium and long wavelengths all together, which if they equally activate all three cones, is the same as if you were viewing a white light. Okay? So it appears white. Here you can see overlapping additive color mixing between the red, the green, and the blue. Where the red and the green overlap, we see yellow because it's equally activating the medium and long wavelength cones, just as if a pure yellow would. And you can replicate colors, this is how they do it on TV, by adding or mixing just 3 different colors of light together. So if you ever disobeyed your parents, got really close to the TV screen, okay, and looked at it, maybe with a drop of water which acts as a magnifying glass, you'll see that there's only red, green, and blue pixels. There's no white, there's no yellow, there's no purple, nothing else, okay? It's just those 3 colors on your TV. And you can, I guess I could do it here, but you can do this with Microsoft Paint in class. Sometimes I've done this. Let's see if I can find Paint. You just go to the color palette, edit colors. You can alter or mix different amounts of red, green, and blue. Okay? So what do you predict if I put in the maximum amount, which is 255, for red and green? What do you predict the color will show? If I put in full red and full green, it's yellow. Okay. Or you can, what would happen if I put in the maximum for all 3? Be white. Be white. Okay. Or you can click on colors and see how much is in each one. That's a pure blue. Here's, what do you predict this one's going to be? A lot of green, but a little bit of blue. Okay. If we move over a little bit, let's see. Equal. Okay. Equal green and blue. Okay. So anyway, that's something you can show in class. Just,
What is it again?
it's Microsoft Paint. It's just part of Windows. Nothing new. And that's how their TVs work, except, okay, for the new ones, which now have a yellow pixel. Okay. So to enrich the color palette. All right. So true or false? Adding red and yellow paint to make orange is an example of additive color mixing.
False.
It's false, right? Okay. Because they're subtracting wavelengths of light. So I know it's always confusing. It's just the way it is. It's as bad as positive and negative reinforcement. But. All right, and the color monitors and TVs do use additive, not subtractive, color mixing. All right, I want to show you this. These are human retinas, which through special techniques have been colored to show which types of cones, the geographic distribution of the different 3 types of cones. These are 3 different people. I think this is in the fovea. The blue ones, note that they're relatively sparse compared to the medium and long wavelength cones, okay, the ones they've colored green and red. Note that if you had 2 tiny spots of greenish light, okay, that fell on 2 different regions of the retina, and one of those regions had, you can't see them here, but there are 4 blue cones there. There's no blue cones in this spot. Would that light appear to be a different color in those 2 different spots of the retina? Yeah, it'd be slightly different. Okay? I bet this one, it would appear a little bit more bluish because you have blue cones in there to respond to it. So I thought you might find that interesting. There's a lot of individual variability in humans.
Could that change by eye as well then?
Yes.
Right? So you cover one or the other and you get
It
a
could.
slightly different.
Yep.
Okay.
Artists are aware that if they have a dominant eye, I had
Really?
a friend who would assess colors with only the right
Oh, by
eye.
squint, like closing. Okay, okay.
There's a super rare condition where you can be colorblind in one eye but not the other. So. All right. I don't know what their brain sees. Their brain probably takes a dominant view of one.
Yeah,
one of-
perceptually.
Yeah, we tend to be one-eye dominant, and the brain tends to kind of ignore the other one.
Oh, yeah.
All right, so we've all done this one, right?
Yeah.
Okay.
Okay.
Okay, you've done this one, but how about let's do this one. Stare at her nose. Okay, stare at her nose. I'll give you a good 30 seconds.
Okay.
Yeah. It's so cool. I like that.
Never ceases to amaze us, right? It's like, wow, it's so neat.
I know. I can't get rid of it now.
So we start to talk about the opponent process theory when we recognize that there's this phenomenon that we call these negative after images, but they don't make sense in terms of the trichromatic theory, right? They don't make sense. If you have three different types of cones, why do we see these opposite colors? Well, here's the definition. I know you're familiar with it, but I do want to start to emphasize now how this is happening in the visual system. Just to give you a little bit more ammunition in case you're asked questions. We know that there's three different channels, essentially, of color. One channel is red-green, okay, meaning that you have cells in the visual system that may be excited by red, inhibited by green, or vice versa. There's both. Okay. We have other cells in the visual system that seem to be tuned to the relative difference in the amount of blue versus yellow. Okay. Maybe excited by blue, inhibited by yellow, or vice versa. And then we have another achromatic, meaning without color channel, which is simply kind of the light-dark system. Is this theory in direct conflict with the trichromatic theory? Is it complementary to the trichromatic theory? Which one's right? This is from a sensation and perception course that I teach. These are the three different types of cones. It is true that there are three types of cones in the retina, no doubt about it. They each respond best to different bands or wavelengths of color, of light, excuse me. The achromatic channel seems to just tell us how light it is, okay, without respect to color. And essentially what happens is that you have the cones that feed onto other cells. Okay. Eventually the ganglion cells leave the retina and go to the thalamus. Okay. These ganglion cells get input from both the medium and the long wavelength cones to essentially say how much light there is. Okay. Just overall light. What we're seeing is a translation, a transcoding from one type of signal or output into a different type in the visual system. The blue-yellow channel, medium and long wavelength cones will excite it. Okay, that's arrows indicated by solid lines, will excite it. Whereas input from the short wavelength cones will inhibit it. So there's a balance, okay, where further excitation, the brain's going to see maybe, in this case, blue. Okay. If it gets toned down, okay, or fewer action potentials, it might say yellow. Okay. In the middle, it's going to be neutral. Okay, there's going to be no color. One way, blue, the other way, yellow. And there's a third channel, the red-green channel, where in this case, red or long wavelengths would excite this pathway, and the medium wavelengths would inhibit it. So we have our neutral point, excitation, we see red, inhibition, we see green. So this is trichromatic here. The transcoding into these three channels really is the opponent process theory. They're both correct. They complement each other. Okay? The input comes from the cones, gets converted in the retina and further up in the thalamus in the brain into these three types of channels. So now, okay, the opponent process theory can explain why we see negative after images. I've never done this before, okay, using this slide, so I'm going to see if it helps. Okay, I'm basically testing out a new way of teaching. Tell me if it works or not. Okay, remember this. 3 different types of cones, each respond best to different wavelengths of light. We also have a red-green channel. We're going to ignore everything from the S cones right now because there's no input from the S cones in this channel. This is the one channel that's red-green. We are in balance, okay? An equal amount of medium and long wavelength input indicates that there is no red or green. We're just in neutral balance right here. And let's imagine that we were staring at a red stimulus, okay, that we were adapting to. Kind of like when we were looking, staring at the girl or staring at the flag, okay? Let's imagine that we are staring at a red stimulus that causes adaptation in the nervous system, okay? Now initially, if we look at the photoreceptor response to these 3 types of cones, when we're looking at a red stimulus, we're going to have a lot of activity in those long wavelength cones. Does that make sense? First one? So a lot of output from those long wavelength cones. What does the nervous system do, okay, over time to a steady input? It adapts, it starts to respond less. Okay? It's almost as if it gets tired. Okay? It's like, I've seen that before. Okay? You don't need to tell me that it's, there's fatigue, adaptation, habituation, whatever you want to call it. There is a turning down of the activity of the red pathway. Okay? But initially, when we first show the red stimulus, we're going to be leaning, the output is, hey, we're leaning red. But then we start to adapt. Okay, in the brain, and we'll still see some red. It's still going to be red, but there's going to be some adaptation. Now, when we show a white background, okay, which contains medium and long wavelengths, okay, which system's fatigued? The red. Okay, how's the green system doing? Oh, we're good. Okay, we're good. So the green system's going to respond relatively more than the red, and so we're going to tilt from seeing red back to, back to green, okay? I don't know. Does that make sense?
Yeah, cool.
So,
I like it.
now, what I wish I would have done is brought this one demonstration that I show. How many of you have seen the dot, okay, that is purple, and it's flashing in sequence in a circle? And as you stare at it, 3 things happen. Okay, number 1, you see motion of a circle moving around the dial, okay, or in a circle, which is an illusion, right? That's it's the phi phenomenon, right? So there's an illusion of movement of a stimulus. Second, what color do the dots turn? They turn green. Okay, how come? It's the opponent process. It's a negative after image. Okay, if you're looking at purple, 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 over time it's going to adapt, and then you're going to, when you take it away, you'll see the opponent color, which is green. You'll see a green dot that's now moving, okay, which isn't moving, okay, and it's not green, and all the other dots disappear, okay, because of the lack of defined boundaries and further adaptation in the visual system. But it's a pretty amazing phenomenon. So this is probably the simplest way to explain it. It is true that adaptation probably occurs at multiple levels. It's probably not the cones themselves. Okay? It's probably either ganglion cells or further up in the thalamus and in the cortex. And the evidence for that is, have you ever done a negative after image with just one eye? Let me go back and let's do the flag one, okay? One eye only, okay? Look at it with one eye. It doesn't matter which eye, just pick an eye. Stare at this black circle in the middle. Now keep staring while I'm going to describe what you're going to do in a minute. It would be obvious that in the eye that you're viewing it with, there will be a negative after image. Okay? Almost guarantee that. Okay? What I'm curious to know is, when I say go, but not yet, okay, I want you to, when it goes to the white screen, I want you to look at it with only the other eye. And tell me if you see a negative after image. Okay, ready?
No.
Okay.
Oh, but then when you open both, it's there.
Well, okay, yeah, it's going to be there if you open both. Now, if you didn't see it in the other eye, what does that suggest about where adaptation is occurring? At what level? Probably just right there in the retina, okay? There are other after images that are more complex, okay, that require more cortical processing just to see the image itself. It does transfer to the other eye. Yeah, it transfers to the other eye. So, not everybody, but some people see it in the other eye, which suggests it's happening back here in the occipital lobe. Okay. So at the level of the rods and cones, which theory fits best? Trichromatic. Okay. Higher up in the brain, we didn't talk about thalamus, but higher up, it's going to be the opponent process that seems to fit better. So they're complementary. They're both accurate theories. And really, really brilliant, actually. All right. So for vision, we don't send out sight rays from our eyes. Okay. Color does not exist in the world, only in our brains. We are sensitive to more than just 3 colors. Okay. Both are correct and complementary. And you know what? Negative after images are pretty difficult to explain. I'll give you that. So. All right, so to conclude, and then we'll have some time for questions. Learn from your students. Okay? You know, if you know they're going to have trouble on a difficult topic as evidenced by poor performance on a test, use that to improve the way you teach, to take more time, whatever it is. Just use that information, and sometimes you'll find that students can explain it to each other better than you can, okay? And be willing to allow them to do that. So we did a little bit of it here today when we had time to discuss with a partner and share your answer. And I have my students do that, and I ask them to provide the reasoning for their answer so that they can not just say, well, I guessed A, okay, but it's A because. And sometimes others can correct faulty reasoning. Give them a lot of opportunities to recall and apply what they're learning. Try to connect it to what they're doing, and once again, I advocate those TOPS unit lesson plans. I plan to share with you my presentation, the parts that I can at least, and I will entertain questions that you have about anything that I've discussed today.